Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize winning lab. This channel has three main goals. One, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. Two, to build a supportive community where we can exchange the most valuable experience based research advice. Three, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.
Okay, let's unpack this. Imagine for a moment you're a skilled mechanic, right? And you're facing this really critical repair on an engine. You know exactly which part is causing the problem. It's crucial, it's right there, but well, you just don't have the right tool in your toolbox to actually fix it. For decades, many disease-causing proteins in our bodies have been exactly like that stubborn engine part, vital to target, but, seemingly undruggable with the tools we had. But here's where it gets really interesting, and this is the key thing. A groundbreaking new study has just shown how we can now, quite literally, glue new tools onto these proteins we couldn't target before, essentially making the undruggable gluable. And this isn't just a small tweak, it feels like a radical, almost philosophical shift in how we even approach drug discovery.
It really is a radical shift, and that's precisely our mission today. We're doing a deep dive into this fascinating paper. It unveils a brand new screening paradigm, a whole new way of looking. And this new method is dramatically expanding the reach of what we call molecular glue degraders. The implications for future therapies, they're potentially immense. It's truly about embracing a fresh perspective on a powerful, actually naturally occurring mechanism inside our own cells.
Okay, so to really get why this is such a big deal, let's maybe set the stage a little bit. You're probably familiar with traditional drugs, the ones that work by blocking a problematic protein's activity. Think of it like, I don't know, putting a cap on a leaky pipe, right? It stops the leak, but the pipe itself, it's still there. But what if you could just remove the entire problematic pipe? Or maybe even better, send it straight to the cellular recycling center.
Exactly. That's the core idea behind targeted protein degradation. It's like specifically tagging an unwanted protein and saying, okay, off to the recycling bin for permanent removal.
And the key players doing this tagging, this sort of cellular cleanup, are these small molecule glues. Now, these aren't just like sticky goo. They're incredibly precise molecules, and they work by hijacking our body's own natural protein recycling machinery. Specifically, they recruit these other proteins called E3 ligases.
Right. And you can think of an E3 ligase as a kind of cellular bouncer. Its job is to spot proteins that need to go, tag them for destruction, and then escort them to the cell's trash compactor, which biochemists call the proteasome.
Makes sense, like a quality control system.
Precisely. And for a while now, one particular E3 ligase called cereblon has been the absolute star of the show for these molecular glues, been super successful, generated a ton of excitement. But, like with any major breakthrough, we've started to bump up against its limitations. We're seeing clinical resistance to drugs based on cereblon glues. And crucially, cereblon can only effectively grab onto a relatively limited number of problematic proteins.
Ah,
So while these cereblon targeting glues were truly groundbreaking, they were kind of like having this amazing super specialized wrench. But it only fits one very specific type of bolt. It worked beautifully for those bolts. Yeah, but the vast majority of these undruggable proteins, they were still just out of reach.
Okay, so that sets up the problem perfectly. If the existing cereblon glues have limitations resistance, narrow scope, what was the big, maybe audacious scientific question the researchers were trying to tackle here? What were they trying to crack open?
Well, what's really fascinating is the core motivation. It came directly from understanding those limitations you just mentioned. There was this urgent need to find orthogonal degrader pathways.
Orthogonal, meaning?
Yeah, meaning totally different alternative pathways. Think of it like this. If cereblon is our main cellular waste disposal unit that we've figured out how to use, we desperately needed to find other disposal units, completely different E3 ligases that we could also learn to hijack. And the researchers hypothesized something kind of counterintuitive. They thought, what if these other untapped E3 ligases already have these hidden latent pockets on them just waiting to be used, but we've just been, well, looking for them the wrong way.
Interesting. So not that the pockets didn't exist, but our search method was flawed.
Exactly. Instead of the traditional approach, which was basically, okay, here's an E3 ligase, let's find a glue that fits it and then see what proteins it can degrade. They completely flipped the script. They thought, what if we try to match the glue not to the ligase first, but to the problematic protein-specific features? And that is the fundamental shift. Moving from a ligase-centric view, where the ligase is the star, to an epitope-centric approach. Here, the unique bumps and grooves, the epitopes on the target protein itself, become the guiding star. It's a really profound change in strategy.
Okay, wow. So this is where it gets really clever then. They totally flipped it. Instead of asking, what can this E3 ligase degrade, they start with the bad guy protein and ask, what unique features on this protein can we exploit to make it degradable? So how did they actually do that? How did they start uncovering these hidden vulnerabilities, sort of latent pockets on proteins we thought were untouchable? How do you even find those sweet spots for a glue to latch onto?
Yeah, that's a great question. I mean, how do you even begin to map these incredibly specific, previously unknown connections between a protein surface and a potential glue molecule? They started with a technique called deep mutational scanning. They used it on the interfaces where E3 ligases normally interact with their natural target proteins. Imagine meticulously mapping every tiny bump, every groove, every subtle curve on a protein surface, almost at an atomic level.
Okay.
They were hunting for the absolute sweet spots, these minimal but critical regions on a target protein. The idea was you could get a glue to engage just right in that spot. It could effectively recruit an E3 ligase and kick off the degradation process. These are the hotspot epitopes.
So they're essentially pinpointing the exact amino acids, the precise molecular shapes that are crucial for that ligase recognition, like finding the specific teeth on a key that make it turn the lock.
That's a great analogy, yeah. It's that level of precision.
That sounds incredibly precise. But, okay, once they knew where to look on these target proteins, once they mapped these hotspot epitopes, how did they actually find the glues to stick there? Finding the right molecule out of potentially billions to fit these unique, newly identified sites, that sounds like a massive needle in a haystack problem.
It absolutely does sound like that. But this is where some truly cutting-edge technologies really came into play, allowing them to tackle that scale. They use something called DNA-encoded libraries, or DELs.
DELs, okay, heard of those.
Right. Think of them as these absolutely enormous libraries. We're talking billions of potential small molecule compounds. But here's the clever bit. Each potential glue molecule in the library isn't just floating around. It's chemically linked to its own unique DNA barcode.
Ah, the barcode.
Exactly. That barcode lets you do incredibly efficient screening. You can basically throw billions of these tagged compounds at your target protein simultaneously. Then the ones that stick, you just sequence their DNA barcodes to figure out what they are.
So you're not looking one by one, but testing billions at once and just reading the labels of the winners? That must speed things up. Massively.
Dramatically faster. Orders of magnitude. It turns that needle in a haystack search into a highly efficient sorting process. But they didn't stop there. To make it even smarter, they brought in AI-guided docking.
AI, of course.
Yeah, you can imagine it like a super smart digital architect.
Yeah.
This AI can not only visualize these billions of compounds, but can also predict, with pretty remarkable accuracy, which specific chemical structures, they call them chemotypes, are most likely to perfectly stabilize the crucial tripartite complex.
Tripartite complex. Okay, break that down.
That's just the key three-way handshake, right? You need the E3 ligase, the target protein you want to destroy, and the molecular glue itself, all bound together tightly in a stable structure. If it's wobbly, it doesn't work well. The AI basically predicts which glue molecules will act like the perfect clamp, holding everything together rigidly. This ensures the target protein gets efficiently tagged by the ligase for removal.
Like a master locksmith predicting the perfect key shape for this brand new custom lock they just found.
That's a very good way to put it, yeah.
Okay, so the predictions sound amazing, the screening is high-tech, but the proof, as they say, is in the pudding. Did this complex process actually lead to effective degraders? Especially for those really tough proteins everyone wants to target?
Absolutely. And the results weren't just promising. Honestly, they were phenomenal. They successfully took their initial hits from the screening and chemically optimized them into nanomolar degraders.
Nanomolar, meaning they work at incredibly tiny concentrations.
Exactly. Super potent. And they did this for notoriously difficult to target proteins like MYC and KRAS.
Wow, MYC and KRAS. Those are huge targets in cancer research.
Yes.
Famously difficult.
Huge. They're major drivers in so many human cancers, and they've famously resisted drug development efforts for decades, largely because they lack those traditional deep pockets that most drugs bind to. So achieving effective degradation for them, that's a massive breakthrough.
Okay, so getting nanomolar potency against MYC and KRAS, that's not just a lab curiosity then. That has real implications.
Oh, absolutely. Nanomolar potency is a fantastic starting point because it means you potentially need very little of the drug to have a significant biological effect. For targets like MYC and KRAS, this offers a real glimmer of hope where there was, frankly, very little before. Of course, there's still a long road to actual therapy involving preclinical testing, clinical trials, all of that. But having highly potent molecules against these previously undruggable targets, it fundamentally changes the game.
And did they have further proof beyond just showing the protein levels went down?
They did. They were very rigorous. They validated these degraders using advanced techniques like proteomics, basically measuring all the proteins in the cell to confirm that, yes, the target protein was specifically degraded and other proteins were largely untouched. And maybe most compellingly, they captured cryo-EM snapshots of these ternary complexes.
Cryo-EM, the super high-resolution microscopy.
Exactly. Cryo-electron microscopy gives you atomic-level images, so you can literally see the molecular glue holding the E3 ligase and the target protein together in that three-way handshake, just like the AI predicted.
Wow.
It provides irrefutable visual evidence of the mechanism. It's like having a high-res photo proving exactly how the lock and key fit together with the glue.
That is compelling. Now, this kind of work, it sounds incredibly complex, groundbreaking stuff, rarely happens in isolation. It must have required huge resources, diverse expertise. Who are the key players behind this deep dive? How did they manage to pull it off?
You're absolutely right. This study is really a fantastic example of modern scientific collaboration firing on all cylinders. Yeah. It wasn't just one lab working alone. It was a major joint venture between three real powerhouses: the Max Planck Institute, the pharmaceutical giant Bayer, and the Broad Institute of MIT and Harvard.
Okay, heavy hitters.
Definitely. And this collaboration brought together this incredibly diverse range of expertise you need for something like this. You had structural biologists, the ones who understand the 3D shapes of proteins at an atomic level. You had cheminformaticians, specialists in handling and analyzing vast amounts of chemical data, like those DEL libraries. And crucially, translational oncologists, the experts who bridge the gap between the lab findings and potential patient benefit.
A real multidisciplinary team.
Absolutely. And beyond just pooling their expertise, they really fostered an environment of open science, which was pretty cool. They used shared cloud notebooks so all the partners could see the data and analysis in real time, which really speeds up discovery. And remarkably, they even provided a no-paywall compound set.
What does that mean?
It means other researchers could get access to some of the key chemical compounds they developed and try to replicate or build upon their findings without hitting financial barriers or licensing issues immediately. It directly tackles a common frustration for scientists just getting access to the materials and data needed to move science forward collectively. It really embodies that spirit.
That's fantastic. Truly collaborative, breaking down some of those usual walls. Were there any specific individuals whose contributions really stand out in this massive effort?
Well, teamwork was key, but yes, some individuals played absolutely crucial roles. Marcus Hartmann, for instance, was central in leading that detailed mapping of the hotspot epitopes, figuring out where to target. Anika Schwarz at EMBL, she led the really complex cryo-EM work that gave us those stunning visual proofs of the molecular handshake.
Seeing is believing, right?
Totally. And then Ryo Tanaka, a medicinal chemist, did the painstaking work of optimizing the initial hits, tweaking the molecules to make them super potent, reaching that nanomolar level. And Clara Ng, an oncologist, performed the critical test to see how well these glues actually worked in tumor models, giving that vital early validation in a disease context. It was a real symphony of specialists.
Amazing. So let's step back again, look at the big picture. What does this all really mean for the future of medicine? How big a deal is this paradigm shift, moving from being ligase-centric to epitope-centric in discovery?
I think it's potentially transformative. It dramatically broadens the entire landscape of what we can target with degraders. You can think of it like creating a kind of modular plug-and-play pipeline for developing these drugs. It's conceptually a bit similar to how CAR T-cell therapies work in cancer immunotherapy, where scientists can swap out different targeting modules, different antigens, to go after different types of cancer cells.
Okay, I see the analogy. Modular design.
Exactly. Now, with molecular glues, using this epitope-centric approach, We can potentially identify these critical epitopes on all sorts of problematic proteins and then systematically search for or design new glues specifically for them. It creates a much more systematic, maybe more predictable, and definitely more scalable way to design these incredibly powerful drugs. It really positions molecular glues to move from being a niche approach to becoming a mainstream therapeutic modality, like antibodies or traditional small molecules.
A new pillar of drug discovery, potentially.
That's the hope, yes. The expectation, or at least the strong possibility, is that within the next decade, this approach will yield first-in-class drugs for targets that have been incredibly challenging for years. We're talking about entire classes of proteins like transcription factors and small GT passes. These are involved in just countless diseases, everything from cancers to neurodegenerative disorders to inflammatory conditions. They've been notoriously difficult to drug with conventional methods because they often lack those nice deep pockets that typical drugs bind to. This new approach offers a clear path forward where, honestly, there wasn't much of one before.
Okay. It sounds incredibly promising, but of course, science is never quite that simple. No breakthrough comes without its next set of challenges. As exciting as this is, what are the main hurdles this technology still needs to overcome on the road to becoming actual widely used therapies? Are these sort of solvable engineering problems, or are there deeper questions still?
Yeah, you're right to ask that. And it's important to be realistic. While the conceptual leap is huge, the challenges that remain are primarily practical, more on the engineering side of things, which is actually good news because those are often solvable with focused effort and ingenuity. The two big ones are probably ensuring on-target selectivity.
Meaning making sure the glue only degrades the protein you want it to degrade.
Exactly. You need to make sure it's not accidentally causing the degradation of other important proteins, which could lead to unwanted side effects. That requires very careful design and testing. And the other major hurdle is optimizing in vivo PK, which is pharmacokinetics.
Ah, PK, how the drug behaves in the body.
Right, how it gets absorbed, where it goes, how quickly it gets broken down or cleared out. Getting that balance just right is crucial for any drug to be safe and effective in a person. It needs to last long enough at the right place to do its job, but not too long to cause problems.
Okay, so selectivity and getting the delivery and duration right in the body. Those sound like familiar challenges in drug development, though.
They are. There's significant challenges for sure, but they're the kinds of problems the field has gotten very good at tackling for other types of drugs. So given the sheer power and potential of this new epitope-centric approach, there's a lot of optimism that the scientific community can work through these.
What an amazing journey through this research. Seriously, from staring down these stubborn, undruggable proteins that have plagued medicine for so long to pioneering a potential new class of drugs that could actually save lives. It's a real testament to how a fresh perspective can unlock so much potential.
It really is. This deep dive highlights so beautifully how a fundamental shift in perspective, moving from focusing on the tool, the ligase, to focusing on the specific part of the problem, the epitope, can absolutely revolutionize an entire field. It's kind of like finding a completely new way to pick a lock, maybe not even using a keyhole, but finding some other vulnerability. And suddenly, countless doors that were previously sealed, shut, just swing open.
That's a great way to put it. So wrapping this up, what does this all mean for you, for anyone listening? What's the broader takeaway from this incredible science story?
On one level, this study offers very real, tangible hope for treating diseases that are currently resistant to therapy. It's about potentially drugging the undruggable, attacking targets we previously thought were impossible to hit. That's huge. But maybe on a deeper level, it also raises this really important, kind of provocative question that goes beyond just molecular biology. In what other areas of science, or even outside of science entirely, might we find similar revolutionary breakthroughs? Where else could we benefit from rethinking our most fundamental assumptions? From shifting our focus away from the established tools or frameworks and instead focusing intently on the epitopes, the specific, critical, maybe even overlooked features of the complex problems we're trying to solve. It's a powerful reminder, I think, that sometimes the most effective solutions don't just come from trying harder with the same old methods, but from learning to see the problem and maybe even the world in an entirely new light.